Tanya, thank you very much for that generous introduction. It's very heartwarming to see so many friends and colleagues here, including faculty from the University of Maryland, some of whom I haven't seen in almost a year. And what I want to do today is to take you on what has been a very challenging, exciting, and rewarding journey for me. About 18 months ago, when I decided to apply for a Jefferson Science Fellowship, I embarked upon a journey from the fascinating, challenging, and beautiful world of proteins. Most of you probably don't think of proteins as being beautiful, probably don't know what they look like, but in fact, I think I'll be able to persuade you that they are indeed very beautiful. To the equally fascinating and challenging and dynamic East Asia and Pacific region, as well as the world of science diplomacy. And what I want to do today is to share some of the experiences that I've had along the way and some of the things that I've learned. So let's begin with proteins. First of all, what do proteins do? Proteins are exquisite macromolecular machines that perform all of the tasks in the cell that is necessary to keep the cells alive and therefore to keep the organism alive. Enzymes catalyze chemical reactions. Membrane transport proteins move molecules and ions through membranes. Motor proteins drive muscle contraction, cell movement, and transport of materials within cells. Signaling proteins transmit signals within and between cells. And the immune system proteins recognize and disable foreign molecules and microorganisms. So as you are sitting there quietly listening to this lecture, all of these processes are going on in your body at a very rapid rate. Just think of those particles popping around in your cells. It's enough to make anyone nervous. So then, what are proteins made of, and how are they made? So proteins are polymers made up of small molecules called amino acids that are hooked together by macromolecular machines called ribosomes, which translate the genetic information in a molecule called messenger RNA, which is of course derived from DNA, into the sequence of amino acids in the protein. There are 23 different kinds of amino acids in the protein, and I've tried to indicate with these arrows that they all have something in common. They have functional groups at either end that are able to connect up with the next amino acid in the chain, but they also have a distinct chemical group which confers unique chemical properties on that particular amino acid. Now, although proteins are linear polymers, they don't exist in this shape within the cell. Instead, they fold up into compact structures, and this folding process is driven by weak interactions between the different functional groups of the different amino acids, plus some of the groups located right on the backbone. Despite their very diverse functions, all proteins have some fundamental physical properties in common, which enable them to perform all of the functions that they are capable of. So first of all, when the protein folds up, it creates a unique surface on the protein, which allows it to selectively and specifically interact with the molecule that nature evolved the protein to interact with. And so this is the surface of the subunit of a protein called NAGS, which you'll be hearing about later. And you can probably see that it has peaks and valleys. It's not a flat surface. But in addition, it has positively charged groups coded in blue, negatively charged groups coded in red, and oily groups, surfaces, coded in green. And so when the protein meets up with its partner, as you would expect, blue is coupled with red, red is coupled with blue, and the oily patches come together because they don't like to be in contact with water. Proteins are also flexible. This folded shape that they assume is not fixed, but undergoes changes depending upon environmental conditions. So here, for example, is the entire NAGS molecule. It's made up of 6 of these subunits, which are coded in different colors. This is the shape of the molecule in the absence of one of the amino acids called arginine, which not by accident is the amino acid that has the most nitrogens of any amino acid. And when arginine binds, it causes the protein to flip its overall structure, just as, since this is baseball season, when a ball hits the catcher's glove changes shape. And then finally, these structures are dynamic. This structural change that I've shown here occurs on a fairly slow for molecules, time scale perhaps tenths of seconds. But the structures are also in constant dynamic motion, and breathing, we call it. And what that does is to allow small molecules to enter the protein and then to get out again. And what I'm going to illustrate that with in just a minute or so is, in fact less than a minute, is a very short video of this particular protein, the potassium channel. And the structure of 2 of its subunits is shown here. Each subunit has 2 long helices. This is one of the structures that the protein tends to form when it folds. 1 short helix up here, and the helices are connected by loops. So here we have 1 subunit, here we have a 2nd subunit, and the potassium ion is able to make its way through this channel between the 2 subunits. And what controls the entry of the potassium ion to the channel is the movement of these loops. Other ions, so potassium is a positively charged ion. Other positively charged ions are not able to enter either because they don't fit the dimensions of the channel. I think I've told you everything that is on these 3. Where the potassium channel is located in the cell membrane of the cell. The cell membrane is largely made of a lipid bilayer, which ions and other charged species cannot get through. But putting the channel in the lipid membrane allows potassium to enter the cell, and potassium is required for many cellular functions. And I've already said that the flexible loops regulate that. Okay, so here we have the lipid bilayer of the cell membrane. So this would be the outside of the cell. This would be the inside of the cell. These are water molecules, and the bilayer is made up of molecules that have long, floppy, oily tails, and then positively and negatively charged groups on the surface to enable the cell to coexist comfortably in its aqueous environment. And then this is the inside of the cell with basically a mirror image of the top half of this structure with, again, fatty tails and charged ionic groups on the surface. Here are the green balls are 2 potassium ions, which would eventually make their way through. You'll see a lot of motion of the fairly fluid fatty side chains here. You'll see occasionally the helices flex, and you'll see a little bouncing around within the channel of the 2 potassium ions, and the water molecules bounce like tennis balls. Okay, so having talked about proteins in general, I'd now like to talk a little bit, very little bit, about how a few proteins are involved in disease. This is actually one of the most important and exciting areas of protein science. So this is work that I've carried out over a period of close to 20 years. It started in Minnesota with a colleague of mine, Dr. Mendel Tuckman, who's a pediatric geneticist, and as you can see, an international team. All of us migrated from Minnesota to this area independently. I came by one path, Mendel came by another. And he is at the Children's National Medical Center at Irving in Michigan. It's an absolutely wonderful place. If you ever have an opportunity, I encourage you to visit it. And so as I say, Mendel is a pediatric geneticist, and what he's been interested in all of his life is diseases of nitrogen metabolism. When we eat food, nitrogen is released when the food is broken down in the form of ammonia, the active ingredient of bleach, and some of it enters the bloodstream. You can imagine that if too much of it enters the bloodstream, that this would not be a good thing. And what it does most prominently is to cause neurological symptoms, which in severe cases will result in comas or even death. To regulate the amount of ammonia in the blood, nature has evolved a biochemical pathway called the urea cycle, which takes ammonia in and converts it to, ammonia is also positively charged, converts it to a neutral molecule containing 2 nitrogen atoms called urea, which is excreted from the urine in the kidneys. The urea cycle is primarily located in the liver. Now, believe it or not, Mendel and I have spent most of this time looking at 2 enzymes in this pathway. The first is an enzyme called OTCase, which helps to drive the pathway converting ammonia to urea. The second enzyme, NAGS, has a different role. You'll notice that it's right up here at the beginning of the transition from ammonia to the intermediates of the urea cycle. And this is an example of a feedback system, which is enormously important in biological and chemical functioning. And so as the arrows indicate, when nitrogen is plentiful, a certain amount of that nitrogen will be incorporated into this amino acid arginine, which I showed you previously. And so arginine is a measure of how much nitrogen is in the system at any given time. NAGS controls the input of ammonia into the urea cycle. And so if you follow these arrows around, the nitrogen released from food turns into arginine, which binds to NAGS through a selective and specific interaction, increasing its activity, and then NAGS produces another small molecule which increases the rate at which ammonia enters the urea cycle. So this siphons off ammonia so that its level in the bloodstream doesn't get above the normal limit in normal patients. If there are mutations in any of the proteins involved in this process, of course it will slow down, and that will cause this hyperammonemia, the raising of levels of ammonia in the bloodstream, and, as I say, very severe consequences for patients. So we began by studying the mutations in OTCase. It turns out that more mutations have been identified in OTCase than in any other enzyme of the urea cycle. So it's the most common cause of hyperammonemia. And we really took it from A to Z, finding the gene, cloning it, producing the protein in bacteria, understanding the protein, crystallizing it, and determining its 3-dimensional structure, and mapping all of those mutations on the protein. We then moved on to NAGS, which has proven to be much more challenging. It's probably associated with the membrane. It's kind of floppy. It's probably stabilized by other proteins. We've now cracked it. We now have taken 5 NAGS proteins from different species through that whole process, and we're hoping that we will get the human enzyme to the same point within the next year or so. Yeah, so this last little addendum just makes the point that when a NAGS is mutated, it will interfere with this feedback loop. Ammonia will not be able to enter the urea cycle efficiently and will instead end up in the bloodstream. Okay, so this is just a pretty picture of most of the structure of OTCase with all of the mutations that Mendel and colleagues identified. The reason there are red and blue balls is that depending upon the severity of the effect of the mutation on nitrogen metabolism, the consequences of a mutation may show up in severe cases right when a baby is born, because it can no longer use its mother's systems to clear ammonia from the blood, so those are shown in red, of course, whereas mutations that don't have such deleterious effects may not show up until adulthood. The patient may be walking around having no idea that they have this particular mutation, and then it often manifests itself under stress, odd kinds of stress. Childbirth is one. Accidents, like car accidents, is another, and eating 24 eggs at fraternity initiation probably also would not be a good thing to do. NAG deficiency and OTC deficiency are examples of orphan diseases. They affect only small populations, which often live in isolated circumstances. The Faroe Islands, for example, has a large distribution of one of these diseases. And there are, of course, many other proteins that have major global consequences. And so I just thought I would show you the 3 proteins involved in 3 of the major health issues of our time: obesity, hopefully not yet, influenza, and senility. So on the left is the insulin receptor. Insulin is a protein hormone that signals cells in the liver to absorb glucose and store it in a polymer called glycogen. And the way this works is that the insulin receptor, you can probably recognize, is another membrane protein embedded in the cell membrane of liver cells with a large external globular domain, a transmembrane region, and then an internal subunit. Insulin binds to this globular subunit on the surface, which transmits a signal through the cell membrane to the beta subunit, which in turn alters its interactions with cell signaling proteins within the cell that says absorb glucose and turn it into glycogen. On the right are 2 proteins that are involved in neurodegenerative diseases that cause senility and perhaps other diseases. So this is the this orange structure is the amyloid precursor protein, which is found in the plaques of Alzheimer's disease. This, more conspicuous in a way, purple protein is called presenilin. It has 8 helices which allow it to loop back and forth through the membrane. Now, as long as the precursor protein stays safely in the membrane, we will not come down with neurodegenerative diseases. However, when proteins, enzymes that are able to clip pieces of protein away, nibble away at both ends of the amyloid precursor protein, it's released from the membrane and seeks out other amyloid proteins to form the amyloid plaques, which we hear so much about. It appears that senilin's role in this is that this particular loop is required in some way for the functioning of this particular enzyme at the intracellular surface of the lipid membrane. Okay, well, moving on from proteins, let's embark to East Asia and the Pacific and science diplomacy. Now, it will probably come to as a surprise to some of the Foreign Service officers in the room to know that some parts of this map were almost as unfamiliar to me when I began working in EAP this fall as the protein structures that I've been showing are to you. So for those of you who also may be geographically challenged, I'm just going to walk through this map. Actually, at the time that I interviewed, I recall asking Tanya and Anderson and Melissa Sweeney how they managed to simultaneously think about 23 different countries that are so diverse. And one of the things that I learned is that it's very helpful to think of sub-regions. So probably obvious to you wasn't so obvious to me 8 months ago. So up here, of course, we have North Asia with 3 economic giants: China, South Korea, and Japan, as well as Mongolia, which is proving to have very large mining resources. Below North Asia, we have the Mekong Peninsula with Laos, Burma, Thailand, Cambodia, and Vietnam. Here we have the maritime states, beginning with Malaysia and Singapore, moving on to Indonesia, the country with the third largest population in the world and the leading Muslim majority country. Timor-Leste, which has only been in existence for 10 years, just had a democratically run election. Then we move on to, I didn't mention Taiwan actually here off China, or Hong Kong as a matter of fact, but then we move on to the Pacific Islands, beginning with the Philippines, Palau, Micronesia, the Marshall Islands, the Solomon Islands, Samoa, and Fiji. And then, of course, down here in the south, we have Australia and New Zealand. One of the things that's very obvious on this map is the amount of water that is present. And this water has a profound effect on many of the geopolitical issues that the Bureau deals with. It's not only the oceans, the Pacific Ocean, but also the major rivers. For example, the Mekong River in the Mekong Peninsula, Yangtze River over in China, and to the east, not part of our bureau, but not too far away, the Indus River in India. Well, this has been a truly wonderful year to be in East Asia and Pacific. I think all of us who came in felt that we had really hit the jackpot when we learned that the United States government had decided to emphasize building its relationships in Asia. As Hillary Clinton said in a very influential article in Foreign Affairs back in November 2011, we are proud of our European partnerships and all that they deliver. Our challenge now is to build a web of partnerships and institutions across the Pacific that is as durable and as consistent with American interests and values as the web we have built across the Atlantic. And so here is Secretary Clinton meeting the Director of the Foreign Ministry of Indonesia just before meetings of ASEAN and EAS. And then at the APEC meeting where President Obama rolled out the Trans-Pacific Partnership, among other things. This has also been the year that the winds of political change blew through Burma. It's been very, very exciting to follow that and the admission of Aung San Suu Kyi to the lower house of the Burma Parliament, together with 43 of her colleagues in the New Democratic Party. This is a hugely challenged region. First, there's its physical scale, 24 million square kilometers. Its huge and rapidly growing population, 2 billion now, predicted to go to 2.5 billion by 2020. Enormous geographic, economical, ethnic, cultural, and religious diversity. Centuries of wars and conflicts, 3 major international wars within the 20th century, important and severe transboundary conflicts, and then internal conflict within various countries, often large scale and with very tragic consequences. Human rights are also somewhat of an issue. Of course, the fundamental human rights are freedom of expression, association, and religion. One of the things I didn't know when I came to the State Department is how actively the State Department is involved in informing Congress, which of course makes funding decisions about the status of human rights within the region. And 3 of the areas that I believe we have to produce reports on are first trafficking in persons, secondly child labor, and third violence against children and women. I don't really need to talk about the expansive and explosive development that's going on, economic development that's going in much of the region, which creates great opportunities but also great challenges. And finally, the subsuming challenge, global climate change, which of course is affecting every part of the world, but is a particular threat, I think, to the East Asia and Pacific for several reasons, which I'll talk about a little later. This will just give you an example of the diversity. This happens to focus on health and economic issues. I decided to include a Western-style democracy, very well developed, Australia, China, which is of course unique. Indonesia, I've mentioned, is huge population and majority Muslim, and then one of the countries of the Mekong Peninsula. Off here to the right are Timor-Leste and also the Pacific Islands, even more challenged than the Mekong Peninsula countries. However, challenges also present opportunities, as is often said, and these are, I think, the major challenges in science diplomacy in this region. Again, they aren't unique to this region, but I'm using this as an introduction to telling you about some of the efforts in science and diplomacy which are taking place around these topics. So the grand challenge is clearly climate adaptation, which, of course, has ripple effects on the environment, especially water issues and biodiversity, energy needs, which in turn propagates to nuclear security. Environmental changes affect food security, and of course all of these have an impact on health. Underpinning all of this is the importance of STEM education and innovation, and we have very robust programs supporting efforts in those areas in this region. So how do we deal with these very large challenges? Well, the answer is here. We work through partnerships. So this slide will show you the national and international partnerships that the State Department is involved in. Again, I didn't understand that all of these partnerships existed when I came here, but most of them and their acronyms have become pretty familiar. So in terms of health, so we work, first of all, very extensively with the other federal agencies, interagency working groups they're called, or IPCs at a higher level. And the principal federal agencies that we work with in terms of health issues are, of course, the Center for Disease Control, Department of Homeland Security, Department of Defense, again, something that I was not entirely aware of, National Science Foundation, and USAID, and we have several people from USAID here today. In terms of the environment, of course, USAID and NSF are also involved, but in addition, we have the Department of Energy, Department of the Interior, the Environmental Protection Agency, NASA, and NOAA. Public-private partnerships are becoming a very big deal as there's concern about the future of the budget, and as corporations become more interested in the opportunities in East Asia and Pacific, there are many opportunities. We also work with civil societies, numerous NGOs, the World Wildlife Fund and the Nature Conservancy, for example. Also the Asia Foundation, which receives congressional funding and is an important player. We've had, of course, bilateral interactions with the countries in the region for many, many years. In fact, at one time, that was the most prominent form of diplomacy. It's become apparent, as I'll say more about later, that in fact bilateral partnerships have many advantages. In terms of science, one of the ways in which goals are established is through joint commission meetings of the leaders of the various technical agencies in both the United States and also the partner country, so-called JCMs. We've just had JCMs with both China on the margins of the SNED meeting and also with Indonesia, and there are more coming up. The goal is to do these on a 2-year cycle. Here are some of the organizations within the region that we work with multilaterally. I'll be showing you a diagram of this in just a minute, but since not all of these are shown here, I'll just step through it. ASEAN is the oldest and has the most comprehensive agenda of any of these multilateral organizations. The Association for Southeast Asia Nations, founded about in the 60s. APEC, the Association, the Asia Pacific Economic Cooperation Organization, East Asia Summit, the Asia Regional Forum, the Lower Mekong Initiative, the Pacific Island Forum, and the Trans-Pacific Partnership, which I mentioned earlier. And then in terms of international organizations, of course, we work closely with several of the UN bodies, the UN Food and Agriculture Organization, World Health Organization, International Atomic Energy Agency, UNESCO, Education, Science and Culture, the World Bank, and the Asia Development Bank, which is a big player in terms of funding. So this is a diagram of the relationship between the countries in the region, broadly defined, and each of the organizations that I just listed. And it'll be helpful to think of this as superimposed upon a map. And so the circles and ellipses that are over here include many countries in Eastern Asia, of course, closer to the United States, and similarly, of course, this particular organization, which I don't hear much about, involves South and Northern Asia. And then, so as you move out in these concentric circles, here we have the members of ASEAN, 5 maritime states and 5 mainland states. ASEAN plus 3 includes the economic powerhouses, South Korea, Japan, and China. The East Asia Summit brings in New Zealand, Australia, and APAC brings in Hong Kong, China, Chinese Taipei or Taiwan, Mexico, Peru, and Chile. Not shown here is the Trans-Pacific Partnership, which will link more North, South, and Central American countries with the East Asia and the Pacific. You can probably read this. It's a little hard to do on an 8 by 11 sheet of paper, but this diagram also includes the foci of the various multilateral organizations. So as I said, ASEAN has a very broad mission that includes all of the top 7, whereas the Asia Regional Forum is focused almost exclusively on security with, of course, a big political dimension. So now moving on to science and technology activities in the region. I've said before that this is a very vulnerable region in terms of climate adaptation for at least 3 regions, as illustrated by this aerial view of the Mekong Peninsula. There are many islands, there are very long coastlines, there are major rivers. As is the case everywhere, there are very high population densities in many coastal regions. The East Asian Pacific region includes 5 of the world's largest megalopolises, and they're all located right on the coastline. There are other, of course, cities located on the major rivers, and these will also be challenged by flooding. And then third, the region has very meager economic, institutional, and technical resources in many areas, and so this makes it difficult to cope with the upcoming challenges, which include disappearing landmass. The Pacific Islanders are just distraught about this, but of course much larger regions along the coast is also likely to disappear. Catastrophic flooding, both from extreme weather and also from too much water running down the rivers. Increased disease susceptibility because the uncontrolled spread of water will create both sanitation issues and also fantastic breeding grounds for insect vectors for insect-borne diseases. Increased energy needs, primarily because of the breathtaking development that's occurring, but also as the temperature rises, of course, there'll be more need for cooling. And then finally, major threats to biodiversity that I'll be saying more about in just a minute. So the next few slides will just describe very briefly what for me have been the highlights in climate, energy, and biodiversity within the last year. First of all, I was just so interested in learning about the Durban Agreement, which took place in the fall, which fundamentally begins to plan is an agreement to begin to plan how to reduce the disparity between developed and developing countries in terms of carbon emissions and the release of gases that produce global warming. This is a wonderful program which one Jefferson Fellow and one triple E Fellow in the office in which I've worked were involved in its early years. As you probably know, the use of traditional cook stoves releases massive amounts of carbon particles and also gases that warm the climate. And they're used by 3 billion people, and they cause 2 billion deaths a year. And so a group of very talented people got behind and visionary people got behind trying to replace the traditional cook stoves around the world with modern cook stoves which have much reduced emissions. This will have the effect of both mitigating climate change and also saving lives and empowering women. This figure is of cook stoves, I believe, in Mongolia. So you can see that these are $2 a piece and can replace the noxious ones that have been in use previously. Third, I was very interested to learn about the tension between the use of hydroelectric power as a source of clean energy and the dams, the effect of the dams that are required to produce it. The dams have of course, very deleterious effects on water flow and therefore the environment downstream. And my first inkling that things were about to change in Burma was when I read that, in fact, Thein Sein had decided to stop the construction of a dam funded by China along the Irrawaddy River, in part because of protests from the people of Burma. This figure, of course, shows a bridge over the Irrawaddy River right in Rangoon. And then finally, energy and nuclear security has been a focus of a lot of attention, partly because of Fukushima, but also because increasing energy needs have created concerns in both Korea and Taiwan specifically as to the extent to which they should use nuclear energy to meet their growing energy needs. There are many programs working to conserve the environment and protect biodiversity in the East Asia and Pacific region, many of them funded primarily by USAID. These programs, the Asia region endangered species trafficking is funded by USAID to the tune of, over the last decade, tens of millions of dollars. And then the ASEAN Wildlife Enforcement Network is a sort of a homegrown program, both with the same goals of combating illegal wildlife trafficking by reducing consumer demand and strengthening law enforcement, regional cooperation, and an anti-trafficking network. Many of the iconic species, as you know, have only a few thousand members still in the wild, the pandas, the rhinos, and the tigers. But there are many other species that are also threatened. For example, a reptile called pangolin, which is a major source of meat. Other reptiles are also used in the same way: birds, flowers, plants, and so on and so forth. So this program is multifaceted. It does a lot of training of both law officers and people who work directly on protection of both wildlife and forests, developing a legal structure, and then also developing ways of using the internet. Actually, is quite a big tool, and other strategies to try to slow down the destruction. This picture, of course, is of a rhino who has had his horn removed, and is clearly pretty badly infected. Both tiger bones and rhino horns, as you probably know, are very important in traditional medicine. They're believed to have magical powers. And then, so this is a mainland program. There is also the Coral Triangle Initiative, which is working to protect coral reefs, fisheries, and food security in 6 regions that form a triangle. One of the strategies is to plant mangroves along the coast to prevent flooding, and the figure at the bottom just shows a relatively mature mangrove grove. Moving on to health. There are three major challenges. First of all, in infectious disease, the big three: tuberculosis, malaria, and HIV. Tuberculosis and HIV used to kill few million people every year. Those numbers are declining, fortunately. Malaria has declined even more sharply, in part as a result of the efforts of USAID. Neglected tropical diseases. These are diseases that are affect millions, billions of people in the underdeveloped regions around the world. Many of them are parasitic worms. Leprosy is one, for example. Leishmaniasis is another. Emerging diseases, there is a constant concern about new diseases that we have no idea of how to deal with emerging from this region for a few reasons. One actually is that it's warmer and therefore genes mutate more rapidly, and so there's more opportunity for new organisms to arise. The avian flu has been in the headlines all year long because of the experiments that produced an airborne flu that infects ferrets. But there are many others to be concerned about. Pandemics that spread around the world, perhaps as a result of emerging diseases. And finally, bioterrorism, which fortunately so far we've done pretty well with. The three major health challenges from environmental factors are air quality, which is a major concern in the very large cities of the region, food and water security, and nuclear security. I've talked about those before. And then non-communicable diseases: diabetes, obesity, cancer, heart disease, and so on and so forth. Highlights in health for me were first the development of a very large consortium called Uniting to Combat Neglected Tropical Diseases. I don't see how you turn that into an acronym, but maybe there's a way. With governmental partners, both the United States, Europe, and Saudi Arabia, corporate partnerships, many of the big international corporations, and then foundations, particularly the Gates Foundation. And the goal is to eliminate 5 NTDs by 2020 and to control 5 others. This would improve the lives of 1.4 billion people. Non-communicable diseases got a lot of headlines right at the beginning of this year as a result of discussions that took place around the United Nations General Assembly. And a resolution was passed that to set the stage for doing more to reduce the spread of non-communicable diseases around the world. Biological threat reduction is a huge activity, and I was actually very comforted to hear that it is such a vigorous effort of the United States government. One component of this is the World Health Organization International Health Regulations, which I think were put in place maybe around 2005, and the goal is to have all countries conform to these health regulations by this June, as a matter of fact. The goal is to make sure that the infrastructure is in place to diagnose, to prevent when possible, to treat, and importantly, to communicate about diseases in every country in the world. Biological threat reduction got a lot of help this year when Congress authorized the cooperative threat reduction dollars, which had previously been used to deal with nuclear and infectious disease issues in the former Soviet Union for biological threat reduction around the world. And I'll show you a slide that expands on that in just a minute. This picture is of the USS Mercy, which is an example of many vehicles and many activities that are involved in East Asia and the Pacific in working to promote health and to help countries deal with the issues that they face. So just to give you an idea of the sort of the magnitude of the bioengagement efforts, this is a list of partners that are involved in this. So there are a bunch of countries, a bunch of organizations in the East Asia and the Pacific that focus on implementing the IHR regulations. There's an initiative in the lower Mekong Delta funded by USAID, the Asian Development Bank, and the Rockefeller Foundation. PACOM is a major player, the Pacific Fleet, with AFRIMS, and NAMRU 2 is an example of a military-to-military collaboration to develop biosurveillance capacity. There are other NAMRUs in other parts of the world. Finally, just a couple of examples of our efforts to promote STEM and innovation activities. GIST is a very successful program that comes out of NIT that works to promote innovation around the world, but has a couple of countries in the East Asian Pacific, Malaysia and Indonesia, as a special focus right now. And so it does all of the things that people who want, or organizations that want to promote innovation do. We have 3 finalists from this region out of the 10 competing for an award from GIST, and we're hopeful that all 3 will win. Next is an exciting program developed by Sandra Laney, who is in the Bureau of Oceans, Environment, and Science, to develop partner programs between U.S. women's colleges and other countries. And part of the motivation for developing this program was because it is thought, and is proving to be the case, a way to overcome the concerns that Muslim parents might have about sending their girls to coeducational schools, and the hope and the likelihood that the parents will believe that women's colleges will provide a more protected environment. Finally, just a couple of slides about science diplomacy in general. I liked the suggestion of Vance Tarikian and Norm Neureiter from the AAAS Science and Diplomacy Center that science diplomacy, there are basically 3 kinds of science diplomacy. On the one hand, there's science for diplomacy, using science as a way to establish a dialogue with countries that may not be aligned with us on some other issues, but are comfortable engaging in scientific discussions and activities because science is relatively value neutral. And also, the countries of the Pacific are very firm believers that science and technology is going to be a big part of their future. Diplomacy for science is the flip side. This is when the State Department basically is needed to help launch large international ventures. So an example of this, both the Coral Triangle Initiative and the USAID ARREST program are examples of this. And then finally, in an ideal world, at least from my point of view, science is an integral part of diplomacy. You don't do science and separately do diplomacy. In every conversation, science and diplomacy are interwoven. And that's the kind of relationship that we have with most of the developed countries. So, for example, the Australian radar telescopes are an example of that sort of thing. When the leaders of Korea, China, and Korea, Japan, and Australia come to the United States or vice versa, the conversations weave back and forth between science and diplomacy. Finally, the last slide are some of the thoughts I've had about the challenges of science diplomacy. First, it requires a deep knowledge of both science and diplomacy. Few of us have both, and so it usually requires partnerships between scientists and diplomats. In every situation, there will be a right balance between science, development, and diplomacy, which are 3 very different approaches. And with people representing all of those points of view in one room together, it's important and necessary to reconcile the 3 agendas. As we move forward with science diplomacy efforts, there are almost always international diplomatic challenges, countries who are maybe a little nervous about us getting too involved in their business and access issues. And to sustain the programs, a lot of work needs to go into building partnerships and institutional supports of the type that I've described, and then working to sustain the resources, the infrastructure, and the funding that is needed to keep them going. So it's a big challenge, but it's also very rewarding and I think very important in the toolkit of American diplomacy. And so I'll stop now. Thanks to everyone, not only the people who are here today, but also the wonderful leaders that I've worked with this year, or worked for this year, starting with Hillary Clinton and her fantastic vision, and Kurt Campbell, the Assistant Secretary for the Bureau, and Tanya, who's been a great pleasure to work with. So I'll stop now and take questions.
Well, I'd like to ask the first question, Norma. I'm Sharon Renko, and thank you very, very much for a wonderful presentation, and also thank you for diving in on so many S&T issues in your region. It really was a wonderful presentation.
Thank you.
My question is about protein science, and I'd like to know where you see protein science going, but also if we could tie that back to EAP region, where are the opportunities for partnerships within EAP for U.S. scientists like yourself who have been working in this arena for so long? Thank you.
So the trajectory of protein science going back to about the beginning of the 20th century was moving from very basic studies that needed to be done just to understand what proteins were. At one time, it was thought that they weren't well-defined protein entities. And then moving through the kind of physical and mechanistic work that I've described, getting a better understanding of how they do what they do. Then the recombinant DNA revolution, of course, gave us a much bigger toolkit. And we now have a pretty broad and deep understanding of how proteins work. As a result, there are two frontiers that are particularly exciting right now. The first is translational medicine, where instead of focusing primarily on studying proteins in labs, it's very tightly coupled to to clinical needs. You'll be very familiar with this from your work at NIH. This has arisen in part because the funders gradually caught on to the fact that putting a lot of money into basic science, as wonderful as it is, didn't necessarily produce the kind of clinical results that we had promised. And so there are great efforts motivated now by dollars to ensure that basic scientists work with clinicians in order to take the discoveries of the lab to the bed. Some people say to the bedside, some people say to the trench, all the way through the life cycle. This work that I described with Mendel was actually a pretty early example of this, and but the work that I described is actually commonplace now in many labs, and moving on to much broader questions. Secondly, synthetic biology offers tremendous opportunities. So as a result of our knowledge of not only proteins but also the nucleic acids, we can now, I can't, but young people can construct little circuits that can be put into cells and basically make cells do anything you want them to do. It's very elegant and very exciting and clearly has great potential. It's also one of these dual use situations. It can be used to do a lot of good. It can also be used to do great harm. And so both the science funding agencies and the security agencies have a great interest in synthetic biology.
So I was going to say that it occurs to me that
you are.
I'm Diana Ma. I'm sorry.
I did not for my benefit, for the members.
I'm a Franklin Fellow in the EAPK at the Korea desk. I was going to say it occurs to me that a nice match between your interests, your scientific interests, and the EAP region is, as you were saying, that there's going to be a lot of tropical diseases. And
yes.
As the climate changes,
yes.
There's more flooding and more water. There are going to be a lot of diseases coming up. And I'm wondering if you were able to, or if you foresee the United States developing partnerships with the region in trying to combat, or try to predict and combat some of these diseases. And if you were able to make any partnerships of your own to continue into the future?
That's above my pay grade at the present time. I've certainly worked to support some of those efforts, but I have not gone out on my own to the region to start one. There are, I was pretty heavily involved in things that are happening in Singapore, which is actually one of our very important bases. So a couple of things happened this year. First of all, there was a READY Center, the Emerging Disease Institute in Singapore, which was funded during the Bush administration, which has now been absorbed into the National University of Singapore. This is actually a terrific thing because it gives it a permanent home and a place within the Singapore system. And so that will be very useful, I think. Secondly, NAMRU 2 has been working throughout the year on a very complex problem, how to move its command group out of Hawaii, where they have been for a number of years, into the region so that they can work more effectively. And that has involved very complex diplomatic issues, requiring close partnership between the Department of Defense and the State Department, and I've participated in all those discussions. Many of the projects that I described deal with exactly what you're talking about, making sure that the country is prepared for, as you say, the increase in infectious diseases, which is very likely to occur, and in fact could potentially affect the rest of the world. But these are very large-scale programs in which require many individuals working as a team rather than one individual sort of going alone. Although, of course, there's always a leader. I hope that's helpful.
No more questions. Let's thank Marla for a wonderful lecture.